So to make a protein, DNA has to go through transcription to turn into RNA, and then the RNA will go through translation to turn into a protein. So at the end of transcription, RNA strands of exons will move out of the nucleus and into the cell. Once the mRNA exits the nucleus, translation will start. So the mRNA enters the cell and will latch onto either a floating ribosome or rough endoplasmic reticulum. If the mRNA latches onto a ribosome, it will stay in the cell. However, if it latches onto rough ER, which is like the highway of the cell, it will move outside of the cell. So in this case, the mRNA latched onto a ribosome. The ribosome is the purple shape. When the mRNA enters the ribosome will begin to read it as it goes through. So the mRNA is going through the ribosome and it's reading it. The ribosome uses tRNA to help read the mRNA as it passes through. The nucleotides that are on the mRNA are read in codons or groups of three. So this would be a group of three, this is a group of three, and this is a group of three. And this is the tRNA right here. tRNA holds what's called an anticodon, and on top of the tRNA is an amino acid. So these three letters here are the anticodon, which is the opposite of the three groups that are on the mRNA, and this down here is the amino acid. There are 20 different amino acids that are unique to each code. Whatever codons are on the mRNA, there will be a tRNA with an anticodon to match it. So, for example, here is the mRNA with the codons, and this is the tRNA that matches it with the anticodons. Once all the tRNA latches onto the codons, the mRNA, the amino acids at the tip of the tRNA will begin to connect. So once all the tRNA is latched onto the codons on the mRNA, these tips right here, these amino acids will start latching together. These little latches are called a peptide bond. Then once all of them are latched on, it will form a polypeptide bond, or basically a protein. That's why it says it's growing a protein, because once they're all latched together, it's a protein. So after translation, the mRNA can become a hormone or an enzyme. This newly formed protein is specific to its task. So whatever protein is made, since there's 20 different amino acids, whatever protein is made, it will be specific to that job. So that's basically translation. Just notice that the codes were switched twice, meaning the code of mRNA at the end of translation is what it was at the start of transcription.